also learned that bones were found, more of them actually over the past week, and they could be just hours away from actually IDing who this skull belongs to.
A skull sits in a laboratory, not in a museum display case, not in an archaeological archive, in an active research facility where scientists have been trying to classify it for decades, and they can't. The bone structure breaks every rule of human anatomy.
This is not the Starchild, but this is the skull that was found laying next to the Starchild on top of the ground.
The cranial capacity exceeds normal parameters. The eye sockets are too shallow. The suture patterns follow sequences never observed in human development. This isn't a deformed child. It isn't a genetic disorder. It isn't hydrocephalus or any known condition that distorts human bone growth. It's something else entirely.
Sure if we've proven that the Starchild skull is from a gray, or if the grays mix with the human race.
And the DNA? The DNA refuses to behave the way human genetic material is supposed to behave. Copper Canyon, Mexico. A teenage girl exploring cave systems near her family's ranch discovers human remains in a shallow burial site.
I've never held the Starchild skull in my hand, but I know that it was found in the 1930s in a cave in northern Mexico by a teenage girl.
Nothing unusual about that. The region had been inhabited for centuries. But she finds two skulls. One normal. One that would challenge everything science thought it knew about human anatomy. The normal skull gets a brief examination and burial. The other skull, smaller, lighter, with an elongated cranial structure, gets passed around the family for decades as a curiosity. A conversation piece, something strange their ancestor found in the desert. Nobody thought to study it scientifically until the 1990s. That's when Lloyd Pye, a researcher specializing in alternative theories of human origins, first examined the skull.
El Paso, Texas, February 18th, 1999. Ray and Melanie Young introduced Lloyd Pye to the Starchild skull.
His initial assumption matched everyone else's. Genetic deformity, probably hydrocephalus, interesting but explainable. Then he measured it. The cranial bone was far thinner than human pediatric skulls, yet demonstrably stronger. The cranial capacity was 1,600 cubic centimeters, 200 cc larger than the average adult human brain. The eye sockets were 15% shallower than the minimum depth required for normal human vision. The zygomatic arches, the cheekbones, were reduced to almost nothing. The parietal region was enlarged beyond any known human population variance. The occipital bone showed fusion patterns that don't occur in human development. Pye brought the skull to forensic anthropologists. Their response was consistent. This doesn't fit our classification models. Not, this is an unusual human skull. Not, this shows rare genetic markers. Not, this represents an unknown population. This doesn't fit. The first genetic testing focused on mitochondrial DNA, the genetic material inherited exclusively from the mother. The results came back human, specifically haplogroup C, consistent with Native American ancestry.
The DNA of her mother's line. So it should be Anunnaki DNA if he's right.
Case closed, said the skeptics. Human origin confirmed. But mitochondrial DNA only tells half the story. It confirms the maternal lineage. It says nothing about the paternal contribution, the nuclear DNA that determines species characteristics, bone development, and cranial morphology.
You add hair, you change the skin tone, you put regular eyes in it. I'm seeing a human being with a slightly misshapen skull.
When researchers attempted to extract and analyze the nuclear DNA, they encountered something unprecedented. Fragments that refused to align with any known human population database. Sequences that didn't match the standard genetic markers used to classify Homo sapiens. Multiple independent laboratories returned similar results. Not contamination, not degradation, not insufficient sample material. Unclassifiable sequences. The language in the lab reports became carefully neutral. Results inconclusive. Comparative database insufficient. Further analysis recommended. But the further analysis never happened. Funding dried up. Laboratory access was restricted. Sample requests were declined without explanation. The skull was returned to storage, not destroyed, not debunked, but quietly set aside. The scientific community had encountered something it couldn't categorize, and rather than expanding the investigation, it chose to stop asking questions. This pattern, Classification failure, followed by archival burial, happens more often than most people realize. When evidence refuses to fit established models, the evidence gets shelved rather than the models getting revised.
It was absolute groundbreaking, pioneering stuff from a man who absolutely knew what he was doing.
The Starchild Skull remains in taxonomic limbo, neither proven alien nor confirmed human, neither extraordinary nor ordinary, just unresolved. The forensic anthropologists who examined the skull weren't fringe researchers or believers in alternative theories. They were mainstream academics with decades of experience in human osteology and cranial analysis. Dr. David Hodges, a forensic anthropologist at the University of British Columbia, conducted the first comprehensive morphological study. His measurements revealed bone density readings that defied standard pediatric parameters. The cranial bone measured 6 mm thick in some areas, half the thickness of normal human skulls. Yet, stress tests showed it could withstand significantly more pressure than typical human bone material. We use established protocols for determining age, sex, and ancestry from skeletal remains, Hodges explained in his preliminary report. These protocols are based on thousands of documented human specimens. This skull doesn't respond to our standard analytical framework. The suture fusion patterns presented the most troubling anomaly. In human development, cranial sutures close in predictable sequences tied to age and hormonal changes. The Starchild Skull showed suture fusion that followed no known human timeline. Some sutures that should close early in childhood remained open.
Guys, I think one of the problems to getting answers to this skull is that it's obviously incomplete. I mean, a large portion of it is missing.
Others that typically stay flexible until adulthood had fused completely. Dr. Marla Weinstein, a pediatric osteologist, attempted to correlate the fusion patterns with known genetic conditions affecting bone development: craniosynostosis, Apert syndrome, Pfeiffer syndrome. None matched the specific combination of features present in the skull. In cases of premature suture fusion, we see compensatory growth in other areas of the skull, Weinstein noted. This skull shows no compensatory patterns. The proportions suggest normal growth within an abnormal framework. The eye socket measurements created additional classification problems. Human eye sockets require minimum depth parameters to accommodate normal vision. The Starchild Skull's orbital cavities measured 15 mm too shallow for standard human optics. Yet the skull showed no signs of blindness-related bone adaptation. Attempts to determine age at death using standard forensic methods produced contradictory results. Dental analysis suggested a child between 5 and 8 years old. Bone density readings indicated skeletal maturity consistent with a young adult. Cranial capacity measurements suggested a brain size that wouldn't fit in a child's skull cavity. We have 3 different aging methodologies giving us 3 incompatible answers, reported the forensic team's summary.
It's an interesting concept, the thought of Greys coming here and abducting people and crossbreeding. But in my opinion, there's just not enough proof.
This suggests either significant measurement error, which we've ruled out through repeated testing, or biological development that doesn't follow human patterns. The team attempted to classify the skull using comparative databases that included thousands of specimens from different human populations, historical periods, and geographic regions. Native American, Asian, European, African, Polynesian, none provided morphological matches. They expanded the search to include specimens affected by various pathological conditions: hydrocephalus, microcephaly, Turner syndrome, Down syndrome, achondroplasia. The skull's features didn't align with any documented condition affecting human cranial development. The final morphological analysis concluded with unprecedented language for a forensic anthropology report. The specimen exhibits characteristics that fall outside the range of normal human variation and cannot be classified using standard taxonomic criteria. Not unusual human, not rare genetic variant, not unknown population cannot be classified. The forensic team recommended genetic analysis as the next logical step. If morphological classification had failed, perhaps DNA analysis could provide answers about the skull's biological origins. They had no idea that genetic testing would create even deeper mysteries. The transition from physical analysis to genetic investigation represented science's attempt to move from observation to molecular certainty. The assumption was that DNA would resolve what bone measurements could not. That assumption proved catastrophically wrong. The first DNA extraction took place in 1999 at a genetics laboratory in Vancouver. The process was standard. Bone powder dissolved in chemical solutions, cellular material separated, genetic sequences isolated and amplified. The mitochondrial DNA results came back clean, human. Specifically, haplogroup C1, consistent with Native American maternal ancestry dating back approximately 13,000 years. The genetics team felt confident they'd solved the mystery. Whatever physical anomalies the skull displayed, the DNA confirmed human origin. The morphological irregularities could be explained as rare genetic variants within normal human parameters.
With what they were looking for, given the design of this creature and the plan for hybridization.
But mitochondrial DNA represents only the maternal genetic contribution. It's inherited exclusively from the mother, unchanged through generations. It tells you about ancestry, migration patterns, and maternal lineage. It doesn't determine species characteristics, bone development patterns, or cranial morphology. For those answers, you need nuclear DNA, the genetic material that combines contributions from both parents and determines the actual biological blueprint of the organism. The nuclear DNA extraction proved more challenging. The bone material yielded smaller quantities of genetic material, and the sequences required more sophisticated amplification techniques. But the lab had processed thousands of ancient DNA samples. This should have been routine. The first nuclear DNA analysis returned results that made no sense. Approximately 85% of the genetic sequences aligned, with human databases as expected, but 15% of the nuclear DNA fragments refused to match any known human population markers, not Native American, not European, not Asian, not African, not any documented human genetic lineage. The lab ran the analysis 3 more times, assuming contamination or processing errors. Each test returned the same result, 15% unclassifiable genetic material. The laboratory contacted Dr. Pat Brown at Stanford University, one of the leading experts in ancient DNA analysis. Brown's team attempted their own extraction and sequencing using different methodologies and equipment. Same result, 85% human, 15% unknown. In 30 years of genetic analysis, I've never encountered DNA that simply doesn't match anything in our comparative databases, Brown reported.
Has led them to the shocking conclusion that there is a hidden code within our DNA.
We're not talking about rare variants or unusual mutations. We're talking about genetic sequences that don't appear to exist in any terrestrial organism. The genetics team expanded their search beyond human databases. They ran the unknown sequences against genetic libraries for primates, mammals, and other vertebrates. No matches. They submitted the sequences to the National Center for Biotechnology Information's genetic database, the most comprehensive collection of known DNA sequences on Earth. The database contains genetic information from millions of species, including extinct organisms, rare variants, and newly discovered life forms. No matches. The unknown 15% of the nuclear DNA appeared to be genuinely unknown. Not rare, not degraded, not contaminated, unknown. Dr. Brown's team attempted to sequence longer fragments of the unknown DNA to provide more context for database matching. The longer sequences only deepen the mystery. The genetic material showed characteristics consistent with complex multicellular organisms, but the specific sequences had no terrestrial parallels. This DNA exhibits the structural complexity we associate with advanced life forms, Brown explained. But the actual genetic code doesn't match anything in our reference libraries. It's as if we're looking at genetic material from a completely separate evolutionary lineage. The implications were staggering. If the unknown DNA represented genuine genetic material, not contamination, not degradation, not processing error, then the Starchild Skull contained genetic contributions from an organism that didn't exist in any known biological classification system. The genetics team submitted their findings for peer review at 3 major scientific journals. All 3 declined publication, citing insufficient comparative analysis and inconclusive methodology. The team offered to provide DNA samples for independent verification. no laboratory accepted the offer. Funding for continued genetic analysis was quietly withdrawn. The DNA samples were returned to storage. The research team disbanded. The official position became, results inconclusive pending further analysis. But no further analysis was ever conducted. The unknown 15% of nuclear DNA remained unknown. Not because science lacked the technology to analyze it, not because the sample material was insufficient, not because the methodology was flawed, because the scientific community chose to stop investigating rather than confront the possibility that the skull contained genetic material from an unknown source. The DNA had behaved exactly as DNA should behave. It had provided clear, replicable results using standard analytical protocols. The problem wasn't with the DNA's behavior. The problem was with what the DNA revealed. The pattern that followed the DNA analysis reveals how the scientific establishment handles evidence that refuses to fit established categories. It's not dramatic suppression or conspiracy. It's something more subtle and perhaps more troubling: institutional disengagement. Dr. Brown's Stanford team had submitted a proposal for expanded genetic analysis in early 2000. The proposal outlined a comprehensive study involving multiple laboratories, advanced sequencing techniques, and collaboration with experts in comparative genomics. The National Science Foundation received the proposal in March 2000. By August 2000, the proposal was declined. Not rejected outright, declined. The review committee's response cited insufficient preliminary data to justify expanded investigation and concerns about sample provenance and chain of custody. But the preliminary data was unprecedented. 15% unknown nuclear DNA using verified analytical protocols. The sample provenance was documented back to the original 1930 discovery. The chain of custody had been maintained through multiple laboratory transfers. The real concern wasn't insufficient data. The concern was too much data pointing toward conclusions that couldn't be comfortably integrated into existing scientific frameworks. Dr. Brown attempted to secure private funding for continued research. 3 biotechnology companies initially expressed interest. All 3 withdrew their support within 6 months, citing strategic research priorities and resource allocation decisions. The DNA samples themselves became increasingly difficult to access. The original bone material remained in storage, but requests for additional extractions were consistently denied. Laboratory protocols that had been routine for ancient DNA analysis suddenly required extensive approval processes and administrative review. Dr. Weinstein, the pediatric osteologist who had conducted the morphological analysis, proposed a follow-up study examining the skull's bone density properties using advanced material science techniques. Her university's research committee approved the proposal initially, then reversed the decision, citing ethical concerns about studying human remains without clear cultural affiliation. But the skull's cultural affiliation had never been in question. The mitochondrial DNA clearly established Native American ancestry. The ethical concerns appeared only after the nuclear DNA results had been circulated within the academic community. Lloyd Pye, the original researcher who had brought the skull to scientific attention, found himself increasingly isolated from mainstream academic collaboration. Invitations to speak at anthropology conferences were withdrawn. Papers submitted to peer-reviewed journals received rejection letters without substantive review comments. The scientific community had made a collective decision that this line of inquiry was not productive, Pye later observed. Not wrong, not fraudulent, not uninteresting, just not productive. The media coverage followed a similar trajectory. Initial reports in 1999 and 2000 treated the skull as a fascinating scientific puzzle. Articles in Scientific American, National Geographic, and Discover magazine presented the morphological and genetic anomalies as intriguing mysteries awaiting resolution. By 2002, media interest had evaporated. Requests for interviews with the research team were declined. Press releases about new findings stopped appearing. Science journalists moved on to other stories. The skull didn't disappear. It remained in storage at a private research facility in Texas. The DNA samples remained preserved in laboratory freezers. The morphological measurements remained documented in research archives. But active investigation ceased. Not because the mysteries had been solved, not because the evidence had been debunked, not because new information had provided conventional explanations. Investigation ceased because the scientific community had collectively decided that some questions were better left unresolved. Dr. Brown's final report, submitted to the genetics laboratory in 2003, contained language that had never appeared in his previous scientific publications. While the DNA analysis has yielded technically valid results, the implications of these findings exceed the scope of our current investigative mandate. Translation: we found something, but we're not going to pursue what it means. The laboratory filed the report in their archives. No follow-up studies were scheduled, no additional funding was requested, no peer-reviewed publications resulted from 3 years of unprecedented genetic analysis. The 15% unknown nuclear DNA remained unknown, not because science lacked the capability to investigate further, because science chose not to. This pattern, technical competence followed by investigative withdrawal, represents how institutions handle evidence that threatens fundamental assumptions. The evidence isn't destroyed or hidden. It's simply set aside, filed away, treated as an administrative matter rather than a scientific revolution. The skull exists in a state of permanent suspension, neither proven extraordinary nor confirmed ordinary, neither classified nor declassified, neither studied nor ignored. Just quietly managed. The Starchild Skull forces uncomfortable questions about how science responds when evidence refuses to conform to established models. What happens when rigorous analysis produces results that can't be integrated into existing knowledge frameworks? The standard scientific method assumes that evidence will eventually resolve into recognizable patterns. Anomalies are temporary. Puzzles waiting for better analysis, more sophisticated tools, or expanded theoretical understanding. The assumption is that mystery dissolves under sufficient investigation. But what happens when mystery persists despite sophisticated investigation? When multiple laboratories using advanced techniques produce consistent results that simply don't fit any known category? The skull represents something more troubling than unexplained evidence. It represents evidence that science has chosen not to explain. 15% unknown nuclear DNA isn't a measurement error. It's not contamination. It's not degraded genetic material producing false readings. Multiple independent laboratories using different methodologies have confirmed the same result. The DNA exists, the measurements are accurate, the analysis is technically sound, but the implications are unacceptable. So the investigation stops, not because the evidence is insufficient, but because the evidence points toward conclusions that would require fundamental revisions to human evolutionary history, genetic classification systems, and our understanding of terrestrial biology. The skull asks a question that science isn't prepared to answer. What if human development has included genetic contributions from sources that don't exist in our current biological databases? That question leads to other questions. How many other specimens have been quietly archived rather than fully investigated? How many genetic anomalies have been dismissed as contamination rather than pursued as genuine mysteries? How many morphological irregularities have been explained away rather than explained? The resistance to classification becomes evidence itself. When multiple scientific disciplines encounter the same specimen and consistently conclude that it doesn't fit their analytical frameworks, the consistency suggests something significant about the specimen's nature. The skull doesn't prove extraterrestrial intervention. It doesn't confirm ancient astronaut theories. It doesn't validate any specific alternative explanation for human origins. But it does prove that science encounters evidence it cannot categorize. And when that happens, the evidence gets managed rather than understood. The broader implications extend beyond this single skull. If science routinely sets aside evidence that threatens fundamental assumptions, how much of our understanding of human history, biological development, and terrestrial evolution is based on selective investigation rather than comprehensive analysis? The skull remains in storage. The DNA samples remain preserved. The questions remain unresolved, not because answers are impossible, because answers are inconvenient. The skull's greatest revelation isn't about its origins. It's about the limits of institutional curiosity when curiosity encounters evidence that demands uncomfortable conclusions. Science prides itself on following evidence wherever it leads. The Starchild Skull reveals what happens when evidence leads somewhere science doesn't want to go. It gets quietly filed away, and the questions it raises get forgotten until someone asks why the investigation stopped. The Starchild Skull remains exactly where science left it, unclassified, not debunked, not proven, not explained, just quietly stored in taxonomic limbo. 15% unknown nuclear DNA, morphological features that don't match human development patterns, bone density readings that defy pediatric parameters, all documented, all verified, all set aside. Science encountered something it couldn't categorize and chose to stop investigating rather than expand its categories. The skull's greatest revelation isn't about its origins. It's about what happens when evidence refuses to fit our models. How many other discoveries wait in storage, unexplained and uninvestigated? What else have we found and quietly decided not to understand?